I think there's so much about cell sorting that should have been automated for a long, long time. I should not have to sit in front of that sorter with all my expertise and all my excitement about science and all my ability to participate in the research process and spend hours and hours watching the drop delay. Now we have a cell sorter that does all that for me, that allows the investigator to put their cells on the sorter and go back to what's exciting about research. I'm Carol Oxford. I'm a staff scientist at Bio-Rad. Before I came to Bio-Rad, I was the core manager for flow cytometry and cell sorting at UC Davis for 25 years. I think one of the best parts about being a core manager is really being able to see the investigators, what their hypothesis is, where they want to go with their research, and really being a part of that. And I think one of the worst parts about being a core manager is sitting and watching a cell sorter sort. Sorting has changed really drastically over the years. I think the biggest thing that changed was the advent of fluorescent protein and molecular biology. These sorts are generally one color, maybe two color sorting GFP and mCherry and some fluorescent proteins. And these sorts are always simple. They're always one color, two color sorts. And these were a huge percentage of the sorting that I did when I worked at Davis. What the S3 does is it fills the niche of the simple experiments. It's a 2-laser, 4-color system that allows the investigator much more access to cell sorting that they didn't have before. I think sometimes in the flow cytometry community, and I know I'm certainly guilty of this at times, is we think about things the way we've always done them, and we try and improve on the same system. And Propel thought completely differently about how to calculate the drop delay. And sometimes that brings to light really new ideas. Sometimes those ideas didn't work. In this case, they worked brilliantly.
I'm Dan Fox. I'm the Director of R&D at Propel Labs. So our idea of optimizing the drop delay calibration process really evolved from turning it on its head. You have particles that are moving at 20 to 30 meters per second. The particles you're looking at are somewhere in the 1 to 10 micron range. So you have these fast-moving, tiny particles that you're trying to detect. Instead of trying to catch these particles in flight, where they're going very fast, let's look instead at where the instrument is set up incorrectly. So when the ProDrop system is calibrating to come up with the optimal drop delay, it starts with a value that it knows is incorrect. So there are particles that are going through into the waste stream, and it can count those and it can detect them. Once it's established that, it can sweep the time delay, the drop delay value, until it finds the optimal point where no more particles are in the waste stream, meaning that they're all deflected. And it sweeps past that, so you build a nice graph. So you see this V showing where it was incorrect, where it was optimal, and then we can pick that optimal value. One of the great things about bringing in people that weren't used to writing flow cytometry software was that they would stop and ask these questions. Well, why do we do it this way? Well, because that's how we've done it for 15 years. Well, is that the right way to do it? And we'd stop and we'd re-question all of these assertions that we had in our head, all these ways of doing things, and we came up with a lot of better ways of doing it. This was totally a rethink on the best way to accomplish the goal of sorting cells.
I think the key to understanding the S3 is its simplicity. But just because it's easy doesn't mean that it's lost any of its power. We have coherent lasers, we have Hamamatsu PMTs, and we have high-speed electronics. You're still going to get the same kind of speed and recovery and sensitivity that you have on the bigger sorters. It's just much simpler to operate. I think the partnership between Bio-Rad and Propel is sort of part of the new paradigm. We combine the strengths of a small company and a big company. Propel brings the engineering and the state-of-the-art expertise, and the large company of Bio-Rad brings manufacturing power, technical support, and service support. I think it's a perfect partnership. Anytime investigators have access to more technology and understand the technology, they're going to do more research. They're going to do more exciting things with the technology. And the applications are endless from that point. So anytime you give them access to more technology, research progresses, and that's what we all want.